see it now.
I hope that you can see amino acids.
I can see it.
Now, Maria, you was attended the previous lecture. Let's start from you, and Lisa will listen, and Lisa will ask the questions, okay? Maria, what is the subject biochemistry?
Oh my God. Is
Uh,
the
okay, if you don't know the answer, I can answer and you will remember it, okay? No problem.
okay.
Mhm.
Mm, yeah, I remember we were talking about this last lecture,
Yes,
and I
biochemistry
said
is the
the
subject which
biology
studies
and
the
chemistry.
biology and chemistry, which studies the reactions in our body. Yes, the metabolic pathways
Yes.
in our. Now let's talk
Mm-hmm.
about the amino acids. What is amino acid? And you can see also the slide. You can relate if you remember something, or you can talk with your own or from slide. Yes, that's why
Yeah, amino acids are the subunit of the proteins, and they are micromolecules that
Mhm,
join together by peptide bonds.
mhm, yes. And what is the protein?
Protein is a sequence of amino acids, and it has four structures, like the primary, secondary, tertiary, and quaternary structures,
Yes.
and they're macromolecules.
Okay. Now let's talk about the difference between micro and macromolecule.
Yeah, like macromolecule is um, heavy in weight.
Mm-hmm.
It contains more than um, mic- micromolecule is from 2 to 20 amino acids,
Yeah, it's
and
small with small way. Yes.
more
Okay,
weight.
and we have a good examples. Amino acid is a micro and protein is a macromolecule.
Yeah.
Whereas, for example, carbohydrates, yes, for example, cellulose is a macromolecule, whereas glucose is a micromolecule. And many glucose together join together while the glycosidic bonds can produce the cellulose, for example, glycogen, which is stored in our body and we can use it during the starvation as a fuel. And glucose is a micromolecule, whereas glycogen is a macromolecule. So it's a good example. Okay. And let's talk about the general formula of amino acids. And Lizzie, please listen to Maria, and after that you will repeat it. Okay?
Okay. Amino acids are organic compounds that contain amine, and carboxyl functional groups, along with a side chain R group specific to each amino acid.
Yes. Once again, it's a amino group, amine. It's a carboxyl group. It's a alpha carbon. It's a side chain. Is it clear, Lizzie?
Yes, it's clear.
Okay, please repeat it or read
Amino
it.
acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each
Mhm.
amino acid.
Yes. So these part of amino acids are the same, and this is specific to each other. These are both are different. Yes, and we have in general more than 300 amino acids in nature. They are presented in plants, in um, fungi, etc. But in the substitution of protein, just 20 amino acids are given. Okay. Maria, please discuss what is given here.
At a physiologic pH 7.4, the carboxyl group is dissociated, forming a negatively charged carboxylate ion and amino group is protonated.
Protonated. What is protonated? It means that one proton is attached to this amino group. Whereas it can donate also Y hydrogen group as a proton, hydrogen molecule, sorry, atom as a proton. Yes. Okay. Now remind me why it's written here physiological pH equals to 7.4.
It is the pH of intracellular
It's our
and
pH, yes, blood pH. But we have a gastric juice which has an acidic pH. We have a urine with an acidic pH. Why it's not given here that it's a physiological pH?
uh,
Physiological in general it means it's a blood pH, yes,
blood pH.
not
Yeah.
other fluid pH. Yes, it's a blood pH. Okay, and what is alpha carbon?
The carbon atom next to the carboxyl group, which is therefore number 2 in the carbon chain starting from that functional group, is called the alpha carbon. Amino
Mhm.
acids containing an amino group bonded directly to the alpha carbon are referred to as alpha amino acids.
Yes, there are different types of amino acids. They are alpha, beta, etc. amino acid, but in that case, we are talking about the alpha amino acids. Yes, okay. Now, which linkage can join together two amino acids?
Peptide linkage or
Yes,
peptide bond.
Lizzie, you will repeat. Listen to Maria, Lizzie, please.
In potence, almost all these carboxyl and amino groups are combined through peptide linkage.
And what is peptide linkage? Please explain. You can use this demonstration.
Is when the one amino acid joins the oxygen with the hydrogen of the next amino acid,
Mhm.
and there is water loss, no hydrogen loss or water lost.
Water, yes.
Water. So
Oxygen from one,
every
hydrogen from another. Yeah.
so then there is two residues. I mean, every amino acid is a residue.
Yes, they are residues, and it's a CO and NH is a peptide linkage between two amino acids. Yes, and it's a one side chain of amino acid. It's a second side chain of the amino acid. Lizzie, please repeat it.
In proteins, almost all these carboxyl and amino groups are combined through peptide linkage.
Yes. One amino acid and second amino acid. They can join together, and oxygen from carboxyl group and two hydrogen from amino group can join together, and they can produce the water molecule, yeah, H2O. And this linkage known as a peptide linkage, and these amino acids are known as an amino acid residues, and this is a dipeptide. Why dipeptide? Because two amino acids are joined together. And how we can calculate how many amino acids are joined together? It's very easy. We can calculate just side chains. R1 and R2 are side chains of amino acids, and we can say that two amino acids are joined together. Okay, I think it's the same. Now let's talk, Maria, about the side chain.
Each amino acid is identical except for its side chain or R group. It is the charge, shape, polarity, and size of a side chain that gives each amino acid its unique biochemical properties. Amino acid side chains are typically grouped into nonpolar, polar, acidic, and basic categories.
Yes. Thank you, Maria. So we can classify the amino acid according to their side chains. Why side chain? Because the rest portion are the same. Yeah, they are identical. But these side chain are different. Yes, for each amino acid. That's why we can distinguish the four different groups. They are non-polar, polar, acidic, or basic amino acids. Yes, let's start from the uh, classification. Lizzie, please listen to Maria and you will repeat this portion again. Maria, please let's talk about the classification.
Side change dictates the role an amino acid plays in protein. Therefore, it is useful to classify the amino acids according to the properties of their side change. Non-polar have an even distribution of electrons, and polar have an uneven distribution of electrons, such as acids and bases.
Yes. Once again, non-polar, they are hydrophobic. They afraid of water. But why? Because they have even distribution of electrons. They have no electrons to donate them with water. That's why they hate water. Yes, they repel water. And a good example of it is the fats or oils. Yes, we can't solve the fat or oil in the water. Yes, they go to the top of the solution. Yes, fats. But we have also another type of amino acids with its side chain. They are polar amino acids, and in that case, these amino acids have an uneven distribution. Why? They have an electrons which can donate water and they can make a hydrogen bond with water. That's why these amino acids are known as a polar hydrophilic. Hydrophilic means that they like water, yes? Okay, thank you, Maria. Lizzie, please repeat it again.
Side chains dictates that role an amino acid plays in protein. Therefore, it's useful to classify the amino acids according to the properties of their side chains. Non-polar have an even distribution of electrons, and polar have an uneven distribution of electrons, such as acids and bases.
Basis, yes, yes. And Maria, let's start from non-polar side chain amino acid. Please classify them. Let's start from glycine.
Glycine, alanine, valine, leucine, isoleucine,
Phenylalanine,
phenylalanine.
tryptophan, yes. Methionine and proline. You can't see it, I
No.
see. Yeah, yeah, okay, no problem. And simplest amino acid. Okay. Uh, okay. Now let's talk about the location of non-polar and polar amino acids in the protein. Maria, please.
So nonpolar amino acids cluster in the interior of the soluble proteins.
Mm-hmm.
You can see polar amino acids cluster on the surface of the soluble protein, and in the
Yeah.
nonpolar amino acids clustered on the surface of the membrane proteins,
Okay.
like the cell
And
membrane.
yes, it's a membrane. Yes, and the membrane are fat. They are lipids. Lipids are fat, and it's a lipid bilayer. And here it's lipids. So they are hydrophobic. They're afraid of water. That's why these are non-polar amino acid cluster in the inside of these lipids. Okay, and they can make a hydrophobic interactions. Okay, okay, and Lizzie, now read what is hydrophobic interaction?
Hydrophobic interactions describe the relations between water and hydrophobes, low water soluble molecules.
Yeah,
Hydrophobes are nonpolar molecules and usually have a long chain of carbons that do not interact with water molecules. The mixing of fat and water is a good example of this particular interaction.
yes. So it's a hydrophobic molecules. Okay, Maria. Now let's characterize the simplest amino acid known as a glycine, please.
So glycine only has amino group. amine group, a carboxyl group, and as a side change, it only has hydrogen,
Mhm.
and I don't remember the other hydrogen group.
Yes, this part is the same, but this R chain is difficult. No, sorry, different, not difficult. It's the simplest. Why? Because the side chain is just one atom of hydrogen. That's why glycine is an optically inactive. Yes, it has no optical properties. Now let's talk about the proline. Why the proline is an exception among 20 amino acids, Maria?
And it has two types of amino group.
Not two
Uh,
types. It has a secondary amino group. Why it's secondary? But because this amino group is participating in the five-membered ring. Yes, it's a part of this five-membered ring. Whereas this amino group, for example, for other amino acids are free. Okay, that's
okay,
why this amino acid is a exception, and this proline can make kinks, turns, disrupt the polypeptide chain, etc. That's why. Okay, it's
okay,
about proline, and if I ask the question why the proline is an exception, you need to find the correct answer that it has a secondary amino group. Okay. Okay, now let's talk about the polar amino acids. Can you hear me? Hello, Maria. Can you talk about the polar amino acids?
yes.
Hmm.
So polar amino acids has zero net charge at neutral pH. That will be 7.4. And they are serine, threonine,
Threonine, tyrosine,
iodine.
asparagine, glutamine, and cysteine.
Yeah.
Okay, what is disulfide bond? Lizzie, please listen and you will repeat.
Many extracellular proteins are stabilized by a disulfide bond. For example, albumin, a blood protein.
Mhm. Can you hear me? Okay. Disulfide bonds. Please, which amino acid can produce disulfide interaction? Which amino acid? Just cysteine. We have two different amino acids with the sulfur inside. They are methionine and cysteine. Where is methionine? I will show you. Okay. Here. It's a methionine. Can you see? It's a sulfur containing amino acid, but it can't produce disulfide interaction with cysteine or another methionine. Why? Because it has a methyl group in it. So it can't make a disulfide interaction. Just amino acid cysteine. Can you see cysteine sulfur
Yes.
can produce the disulfide interaction. Why? Because it can donate, it can release this hydrogen and make the disulfide interaction with another cysteine, not with another methionine. Just two cysteines can make a disulfide interaction. Okay? And this disulfide interaction can stabilize the tertiary structure of the protein. We discussed it today. Okay, okay. Let's leave it because it's. I think. Do you have a question about the abbreviations, Maria? Lizzie, you
No.
can read it with your own because I think it's a very easy portion. So I don't
Yes.
want to waste the time. Now let's talk about the optical properties of amino acids, Maria.
Um.
Amino acids are optically active, yes. They can produce two different types. They are D
Oh,
and L forms. And what is optical properties? They can make the mirror images of each other. They can rotate, and this rotation can cause that this amino group is given in the one or another side of the molecule. Yes, so there are two different types, D and L, dextro and levo rotation, it's known like that. So in that case, we can see that just L amino acids are given in our body. Just L form of amino acids. Yes. So they are known also as an stereoisomers, enantiomers, optical isomers, or etc. And Lizzie, please read what is isomer?
Isomers, each of two or more compounds with the same formula but a different arrangement of atoms in the molecule and different properties.
Yes, it means that they have the same formula but different structure. Okay, and here we have the polypeptide chain. And Maria, please explain what is polypeptide chain.
each polypeptide chain is a series of amino acids joined together in a specific sequence. Because each amino acid has a unique R group, a particular amino acid sequence creates a polypeptide whose function is dependent upon its chemical structure.
Okay, and what is amino acid residue, Maria?
Is is the the attachment of two amino acids losing water?
Yes, okay. And I want to ask you, okay, let's keep it. I want to ask the questions. Why the proline is an exception?
Because it has a secondary amino acid structure.
Amino group.
Amino group. Yeah,
Oh,
I don't have it too clear. Like proline has amine, and that's a secondary amino group.
yes. Okay, let me show the proline again. Okay,
Okay.
here's the proline. Yes, it's another amino acid. It's alanine. Yes. So
Mhm.
pay attention. This amino group is free.
Mhm.
And when it's ionized, it consists H3. But
Mm.
it's an ionized form, it consists H2. Why? Because this nitrogen is a part of this ring. Yes, this amino group is participate to produce, it participates in the building of the side chain.
Oh,
Okay?
oh, yeah, yeah, yeah.
Whereas it's a free, it's not participating in the building of the side chain. Please pay attention here. It's a
Mm.
proline. Where is proline? It's a proline. And they are the rest amino acids. Yes, these
Oh,
amino groups
yeah.
are free. Yes, you can see these parts are the same for all amino acids, but it's an exception. Why? Because it's a part of the alpha carbon, and at the same time is a part of the side chain. Okay?
Yeah, I got it. So the side change is attached to the amine group of the amino acid, so it's not like free. It's attached to
Yeah.
the side change. Okay, I got it.
Okay, Lizzie, is it
Thanks.
clear?
Yes, it's clear.
Okay, Lizzie, you just need to read it once and you will got it because I think it's simple material. Okay, now I have another question. Why glycine is the simplest amino acid?
Because it only has like a hydrogen as a side change, hydrogen
Mm-hmm.
atom.
Yes, you're right. Why? Okay, which amino acid don't have optical properties?
define non-polar?
No. Glycine, just glycine. Why? Because after the
Glycine.
rotation, it looks like the similar. It has no mirror images. Why? Because it has no side chain. Yes,
Okay.
that's why. That's why. A glycine is an optically inactive.
In
And what can join two amino acids together?
peptide bonds, peptide
Peptide
link.
bond. Yes, you are right. Now let's revise the proteins, and that's it. Okay, Maria, let's start.
The 20 amino acids commonly found in proteins are joined together with a peptide bond. The linear sequence of the linked amino acids contains three-dimensional shape.
Yeah, okay. And let's start from the primary structure. Yes, we have four different structures of amino acid: primary, secondary, tertiary, and quaternary. And what is the primary structure of the protein, Maria?
It's just a simple sequence of amino acids, like a long chain.
Okay, and which bond is given in the primary structure?
Mm,
Just a peptide bond, yes. Why just a peptide bond? Because the amino acids they join with this peptide bond, yes.
oh yeah. Like
Just peptide bond is given on the primary structure of the proteins, yes. But we have also the secondary structure. There we have a peptide bond and also we have a hydrogen bond. Let's talk about the tertiary structure. In the tertiary structure, we have the peptide bond from the primary structure, hydrogen bond from the
hydrogen,
secondary structure, and
disulfide.
also we have ionic disulfide, hydrophobic, and hydrogen bond from the tertiary structure.
Okay,
And combination of the secondary and tertiary structure and many globular proteins together is a quaternary structure.
okay.
Okay, okay. Now, once again, Lizzie, now let's talk about the peptide bond.
In proteins, amino acids are joined covalently by peptide bond.
Mhm. Yes, it's a covalent bond. Yes, it's a rigid, strong, and hard bond. Okay, now Maria, let's talk about the direction of the peptide chain.
So the free amino end terminal of the peptide chain is graded to the left and the free carboxyl end C terminal to the right. So therefore all amino acid sequences are read from N to C terminal to the peptide.
Okay, now explain with your own words. What is the direction of the chain?
So I understand that free amino acid you start to read it from the N terminal, which
Yes,
is
you
left
need to understand it.
to right.
Listen, please listen once again. It's a direction. Its polypeptide starts from N. Why N? Because it's amino group here. N, nitrogen. And it's end with C terminal residue. Why C? Because this carboxyl group is given at the end of the um, polypeptide. And this C means that this is a carbon, yes? So direction comes when we want to name this polypeptide chain. We need to start from the N residue. For example, alanine, valine, leucine, isoleucine, etc. till the end, glycine, for example. Yes. So we need to start with from N terminal to the C terminal. Okay. And
Okay.
what is it? It's about amino acid residue. Okay, it says. And let's talk about the name of the polypeptide. Yes, it's a three peptide. Sorry, three peptide. Yes, valine, glycine, and leucine are joined together with a peptide bond. And we need to start from valine. Why from valine? Because it's an N-terminal amino acid. Okay,
Mm-hmm.
and leucine is a C-terminal amino acid. And please read it.
when a polypeptide is named, all amino acid residues have their suffixes in, an, ik, or ate changed to il, with the exception of the C-terminal amino acid.
Why it's exception? Because it's the last one. And you can see these
Mm,
suffixes is changed with YL, glycine with the YL, and leucine is the same. Why? Because it's a C-term, C-terminal amino acid. It's the last one. Is it clear,
okay.
Lily, for you?
It's clear.
Clear. What about you, Maria?
Yeah, it is clear.
Is clear. Okay. You can read it about the peptide bond, and let's talk about the secondary structure of the protein.
The polypeptide
Mhm.
backbone does not assume a random three-dimensional structure, but instead generally forms regular arrangements of amino acids that are located near to each other in the linear sequence. These arrangements are termed the secondary structure of polypeptide.
Yes. So in the first time we have a polypeptide with a peptide linkage. In that case, we have a polypeptide with a hydrogen bond. But in that case, hydrogen bonds between the peptide link hydrogen and peptide bond oxygen. Yes. Can you explain what is alpha helix? Maria, can you talk about alpha helix?
The alpha helix, beta sheet, and B bend turn are examples of secondary structure of proteins.
Yes. When we have the alpha helix, it means that just one polypeptide chain is coiled, and there is a bond between the first and fourth amino acids, oxygen and hydrogen of the peptide chain. It's a core of this polypeptide chain, alpha helix, whereas this is a side chain. So this side chain not participate in the bond formation. They are extended outwards of this alpha helix, yes? And we have also, okay, here's the hydrogen bond. And these hydrogen bonds are parallel to the coil, yes, to the alpha helix, to the axis. Okay. Okay, Lizzie, please read it. Lizzie, are you here? Please.
Alpha helix is stabilized by extensive hydrogen bonding between the peptide bond carbonyl oxygens and amine hydrogens that are part of the polypeptide backbone.
Yes. So alpha helix is stabilized via the hydrogen bonds between O peptide bonds oxygen and peptide bonds hydrogen. But this bonding occurs between the first and fourth amino acids. So the second can link with the fifth, the third can link with the sixth, fourth can link with the seventh, etc. So these bonds can stabilize this coil spiral form. Okay. Maria, please explain what is given here. It's the same, but I want to listen how you can explain this slide.
Mm, so alpha helix, hydrogen bond, they are. Mm, this is the parallel. No,
Mhm.
parallel.
Between the first and fourth amino acid. Linkage, hydrogen link between the first and fourth amino acid. Yes, second and fifth, then third and sixth, etc. Yes,
Okay.
how it can produce the hydrogen bond from N terminal to C terminal. You can see the arrows and which can give the direction of the polypeptide chain.
Okay.
Okay. amino acid that disrupt an alpha helix. Lizzie, please read it.
Proline, its secondary amino group, is not geometrically compatible with the right-handed spiral of the alpha helix. A large number of shared amino acids by forming ionic bonds.
Mhm.
Amino acids with bulky side chains, branched amino acids.
Yes. So these are amino acids which can change their direction or can disrupt the alpha helix. Proline. Why proline? Because it's an exception with this secondary amino group. Also, there are a large number of charged amino acids which can make the different bonds, yes, and they can change the shape of the alpha helix. Also, there are amino acids with a huge side chains which can change, of course, the alpha helix structure. On branched amino acids, it's the same. It's such as tryptophan, for example, is a branched amino acid. Now let's talk about the beta sheet, Maria, please.
The beta sheet is another form of secondary structure in which all of the peptide bond components are involved in hydrogen bonding.
Hmm. Yes, it means that another form of secondary structure, which can produce the hydrogen bonds between the peptide bonds hydrogen and oxygen again, but in that case, it looks like a sheet, yes? It's just one amino acid, it's just one polypeptide chain or two polypeptide chains, and these bonds, these hydrogen are bonds are perpendicular of this polypeptide backbone. Whereas in the first time when we talked about the alpha helix, these hydrogen bonds were parallel of these polypeptides. Yes. In
Oh,
this case, they are perpendicular. Yes. And let's compare alpha and beta forms of secondary structure. Lizzie, please read it.
Unlike the alpha helix, beta sheets are composed of two or more peptide chains or segments of polypeptide chains. In beta sheets, the hydrogen bonds are perpendicular to the polypeptide backbone.
Whereas here, the hydrogen bonds are parallel. Okay, it's the same. Okay, Maria, explain it, please.
so parallel and anti-parallel sheets. A beta sheet can be formed from two or more separate polypeptide chains or segments of polypeptide chains that are arranged either anti-parallel to each other or parallel to each other. So in the parallel beta structure is from amine free to C terminal, and then from C terminal to another C terminal of the next polypeptide chain.
Maria,
So
when the polypeptides have the same direction, it's a parallel. When the polypeptides have a different direction, it's an anti-parallel. Okay?
okay, when polypeptide have a different direction, is anti-parallel.
Yes.
Okay, and when they are the polypeptide are in the same direction, is parallel.
Yes.
Yeah.
Okay, once again, explain it.
So here we have a parallel beta sheet, and you can see that um, parallel have the same direction, C terminal to N terminal. In the other change is as well the same thing, C terminal and the N terminal. And in the anti-parallel beta sheet, you can see that they are not in the same direction. They are C terminal to N terminal, and then N terminal to the C terminal of the two polypeptide chains.
Okay. I think it's clear for you. Lizzie, is it clear?
Yes, it's clear.
Okay. Lizzie, if you have any questions, you can email me also during the reading, okay? And we have an inter and intra-chain bonds. Please, let's feel the difference between them, Maria.
So when the hydrogen bonds are formed between the polypeptide backbones of separated polypeptide chains, they are termed interchain bonds.
Mhm.
A beta
Interchain bonds
sheet
between them, yes, but
A beta sheet can also be formed by a single polypeptide chain folding back on itself. In this case, the hydrogen bonds are interchange bonds.
intra-chain.
Yeah, intra, and the other one is inter. Okay.
Is it clear or not?
Yeah, it is
It's
clear.
a different, separate polypeptide. It's a one polypeptide folded in many times, okay?
Mm-hmm.
Between the different polypeptide, between the separate proteins, the bonds are interchain bonds, but intrachain are inside the one polypeptide chain.
Okay.
Okay, and what is the beta band?
Beta bands reverse the direction of polypeptide change, helping it form a compact globular shape.
Yes, it's a polypeptide chain, it's a turn. And this turn can cause via the different types of the amino acid. Please, Louise, help to Maria, and now you read this part.
Permits the change of the direction of the peptide chain to get a folded structure. It gives protein globularity rather than linearity. H bond stabilize the beta turn structure. Proline and glycine are frequently found in beta turns. Beta turns often promote the formation of antiparallel beta sheets.
Okay, is it clear?
Yes.
Okay, it's about motifs. Please, Maria, what is motif? Please explain this part.
Structural motifs are short segments of protein 3D structure which are spatially close but not necessarily adjacent in the sequence. Structural motifs may be conserved in a large number of different proteins.
Yes, motifs is a part of the protein which has the different shapes. Yes, they can be parallel, anti-parallel. It's a parallel part. It's a beta parallel. They are anti-parallel. This is a parallel which consists of alpha, beta, and alpha subunits, etc. This is a motif. So we talked today about start from the tertiary structure, and you need to prepare your material from the tertiary structure till the enzymes. Yes, and the enzyme pages you can find on the enzyme slide, last slide. Yes, as it was given here. On the last slides, you can find the pages which you need to read and prepare for the seminar and for the exams. Okay, and I have just one request. Please try to study from book, not from presentation, because I will prepare the exam questions from the book. Yes,
Okay.
they are similar. Presentation and uh, book are similar, but anyway, uh, you need to study the material from the book. Okay.
Okay, thank you.
Okay, have you any questions?
No, not so far.
No. Lizzie, what about you?
Everything is clear, and thank you for my problem.
Sorry, sorry.
Thank you
Okay,
for
okay,
today's
Lizzie,
and
I will share the meetings. Maria, do you have our recorded meetings?
Yes, I have them. I can
Okay,
share
you
it
have
with
them. But
Lizzie.
you can share it with Lizzie, okay? Lizzie, Maria can share it for you. Or if you want, I can share. But Maria, please help me and share the presentations and recorded meetings with Lizzie, please, okay?
Okay.
Okay. Thank you. Good luck.
Okay.
I will
Have
see
a
you in
nice
the
day.
next one.
Thank you.
Okay, you too. Bye bye.
Bye.
Goodbye.
Marita.